Welcome students to another class of botany. In the previous class we have discussed about the different structures, that is structure of virus, and we have discussed in detail about the structure of tobacco mosaic virus as well as bacteriophages. Now in this class we will see how virus multiplies. We will see the multiplication of bacteriophages. As virus is an inert particle outside the living organism, so here these viruses are obligate intracellular parasites. Intracellular means inside the cell. Obligate intracellular parasites. That means they compulsory need to invade the host cell in order to multiply, in order to increase in number. They need to invade a host cell. That host cell can be either plant cell or animal cell or the bacterial cell, but it is obligatory for a virus to invade a host cell. Okay, so since viruses are obligate intracellular parasites, that is, it is compulsory for the virus to invade the host cell. So the multiplication does not take place until and unless the virus invades the host cell. The entry and exit of the virus inside and outside the cell, host cell. The entry of the virus inside the host cell or the exit of the virus outside the host cell. It may be it may be different. Entry and exit may be different, but the mechanism of multiplication will be the same. The mechanism of multiplication of the virus will be the same. That is, it will invade, the virus will invade the host cell. They take over the machinery of the host cell and command the host cell to multiply their genetic material. That is, the viruses, they invade. The mechanism of virus multiplication is they invade the host cell, they take over the machinery of the host cell and they command the host cell to prepare or synthesize the genetic material of the virus. So that mechanism is altered by two cycles. The multiplication of viruses or multiplication of bacteriophages can be by two types. One is lytic cycle and another is lysogenic cycle. Viruses can multiply or increase in number with the help of these alternating mechanisms. That is, one is the lytic cycle and another is the lysogenic cycle. Lytic cycle ends with lysis, that is breakage. Lytic cycle ends with the lysis of the host cell. The host cell, once the virus infects the host cell, then what happens after its multiplication is done? After the multiplication of the viral genetic material is done, the cell will be destroyed. The bacterial cell or the animal cell or the plant cell will be destroyed. So in lytic cycle, the cell in which the virus takes shelter, virus invades, the cell will get destroyed or broken down. So that is lytic cycle. In lysogenic cycle, the host cell does not get broken. The host cell does not get broken. It remains alive. The host cell remains alive. In lytic cycle, host cell gets damaged or destroyed or died, and here in lysogenic cycle, the host cell will be alive. It will not break. Coming to this one term is there, virulent phages. Phages that attack bacteria. Phages or the viruses that attack bacteria, bacteriophages that attack bacteria such as E. coli and causes the breakage of the cell. Some viruses are breaking at the end, they are breaking the cell. Some are not breaking the cell. So the viruses which breaks the host cell, they damage the host cell, completely destroy the host cell. So such bacteriophages are called as virulent phages. Such bacteriophages are called as virulent, that is infective phages. Now we will study in detail about this lytic cycle as well as lysogenic cycle. First we will study in detail about lytic cycle. Lytic cycle takes place in following stages, following steps. The first step is attachment, the second is penetration, third is biosynthesis, fourth is maturation or assembly, and the fifth one is bursting, that is bursting or release or lysis, lysis of cell, lysis of cell. Lytic cycle takes place in following steps. The first step is attachment, the second is penetration, the third is biosynthesis, the fourth is maturation and assembly, and the fifth is lysis of the cell and release of the new particles, new virus particles. So let us study in detail about all the steps one by one. The first is attachment. The first is attachment. For example, this is a bacterial cell. This is a bacterial cell, and here this is the bacteriophage. Now the tail fibers are there here. The tail fibers, they attach itself to this cell wall of the bacteria. So here tail fibers help in attachment or adsorption, attachment of the phage to bacterial cell. The first thing which happens is this bacteriophage attaches itself to the bacterial cell on the cell wall site, on the site of the cell wall. Okay, after it gets attached, it contracts. This one, this tail sheath contracts. The next comes the penetration. After attachment, attachment of the bacteriophage to the bacterial cell or host cell is done with the help of the tail fibers. Is done with the help of the tail fibers. These are the tail fibers and this tail pin. So what happens in the next stage is penetration. In this now, this sheath, the tail sheath contracts, and because of this contraction now, this pin get pressure. This on the pin, pressure will be put, and this pin will make a hole, that is it will inject itself inside it. So the tail sheath helps in penetration by contracting tail core and is driven through the cell wall. Because of the contraction, this is the tail sheath, this is the that is base plate, these are the pins, okay, and this is the bacterial cell. So when this tail sheath contracts, so when the tail sheath contracts, it puts pressure on the tail pins, and this tail pins, what they drive through the cell wall gets disturbed. Now, so through the cell wall, the capsid of, now after this one, after they have driven in, after the tail sheath has been contracted, now here whatever the genetic material, this is the genetic material, inside this genetic material is present. Now the genetic material from the head passes through the tail and enters inside the cell. This is the bacterial DNA. This is the bacterial DNA and this is the viral DNA. This viral DNA which was in the head has been transferred through this tail into this bacterial cell by the tail pins and the tail fibers. Now, why that is genetic material is inside the bacterial cell. What is left? Only the capsid is left. The capsid cannot enter inside the bacterial cell. It will remain outside the bacterial cell. Only genetic material will get injected inside the bacterial cell. So now capsid will be remain outside, fall somewhere. So that part, that capsid is called as ghost. The capsid without genetic material, because genetic material has now entered inside the bacterial cell. So, the capsid without bacterial, yes, the capsid without this genetic material, this capsid is called as ghost, it falls down somewhere. Okay. Now, the genetic material is inside the bacterial cell. Now, this virus, this genetic material command the bacterial cell to prepare the genetic material of prepare the different parts of the virus. This genetic material, it will give command to the bacterial DNA to start the synthesis of the parts of the virus. Now, a new complete virus will not be formed. As soon as it injects the genetic material of RNA inside the bacterial cell, now here biosynthesis will take place. The third step is the biosynthesis. In biosynthesis, now individual particles like capsid, head, that is capsid, genetic material, tail fibers, tail pin, all these individual materials will be formed. For example, this is the bacterial cell. So, inside this na head will be formed, few head particles will be formed, then few tail particles, that is tail particles will be formed. Then we have this genetic material, copies of genetic material will be formed. Then tail fibers, all these individual parts will be formed during this biosynthesis. First is attachment of the bacteriophage to the bacterial cell, then penetration of the genetic material inside the bacterial cell, then after penetration the viral DNA will command the bacterial DNA to make the copies of the individual parts. Small, that is parts will be formed, many heads, many tails, many number of RNA genomic copies, many number of genomic copies will be formed. Now, after this is formed, now after the injection for some period, we won't find the total complete virus. We won't find complete virus. So, next stage is maturation stage. After injection, it will take some time to prepare the individual parts. So, that phase in which individual parts are formed and we doesn't see a complete virus, that phase is called as, or that period is called as eclipse period. The phase from in which, from here this one individual parts are formed, not complete virus is not formed. So, from individual parts till a complete virus particle is formed, that phase is called as what? Eclipse period. That phase is called as eclipse period. Now, here individual parts were formed. Now, what happens in the next stage? The individual parts will assemble. In the next stage, the individual parts will assemble. See, tail, head, tail fibers, all of them are assembled. Here, they were just produced in haphazard manner, they were not assembled one. Just production was taking place. Synthesis of individual particles was taking place. Now, all these individual particles have been assembled into a new virus particle. So, that is called as maturation. And for this assembly of this all viral particles into a new virus particle, so it will take time. So, that time is called as the eclipse period. That time is called as eclipse period. Now, the virus particles are fully formed. Virus particles are fully formed. They are ready to come outside. So, what happens? The bacterial cell, this bacterial cell will produce an enzyme called as lysozyme. Bacterial cell will produce an enzyme called as lysozyme. That lysozyme, for example, this is the bacterial cell, with all those newly formed viral particles. Okay. So what happens? Because of the production of this lysozyme enzyme, this plasma membrane will get dissolved. The plasma membrane of the bacterial cell will get dissolved and the virus will come out. The virus will come out. So this stage is the last stage, which is called as lysis, in which the viral particles, which are now called as virions, that is newly produced viral particles, newly produced viruses, those virions will be coming outside by the dissolving of plasma membrane. The bacterial cell will produce an enzyme called as lysozyme. That lysozyme will dissolve the plasma membrane, and with the dissolving of the plasma membrane, the new viral particles, which are virions, they come outside. They come outside. So that is the newly produced virions which comes out from the lysis, by the lysis of the cell, that is called as burst size, and it will be the newly formed particles will be nearly 50 to 200. It will be nearly from 50 to 200 new particles will be formed. So now at the end, the bacterial cell has been destroyed. Only thing which is present is the new particles of viruses. So that is why we say at the end of the lytic cycle, the cell, host cell will break down. Host cell will die or break down, damaged, and the new particles will be formed. So this was about the lytic cycle. That is, it takes place in the, that is, few steps. First is attachment of the virus to the cell. Then after attachment, the genetic material of the cell is penetrated inside the bacteria. Then the bacteria, that is the virus RNA, will command the bacterial DNA, or the viral DNA will command the bacterial DNA to synthesize the parts. After the synthesis of individual parts of the virus, now all the virus parts will gather and form a complete virus. After the formation of complete virus, the bacterial cell, with the help of the lysozyme enzyme, it will get dissolved. Plasma membrane of the bacterial cell will get dissolved and the new particles will come out, and that is the last stage, that is the lysis or the burst and release. So in this way, new particles will come outside. Then again a new cycle will start. That newly formed particles will again infect the other bacteria. Then continuous cycle will be formed. So this is called as the lytic cycle. In the next class, we will take the lysogenic cycle.